Good afternoon, dear students. Thank you for the invitation. I would like to take this opportunity to present to you the current biotech capability of our university, which I divide into three aspects. The first one is the biotech education, which I will discuss with respect to the biotech track. Second is on the research aspect, and third is on the service to community. And so I would like to begin by going back in 2014 when I returned to the university after several years of pursuing my doctoral degree in molecular biology and biotechnology. And up to this point, so from 2014 to the current time, I have mentored a total of two batches of biotech students. The first batch is composed of 8 students, and then the second batch is composed of 12 students. So, with respect to the other tracks, there are less takers in the biotech track, largely because I think biotech as a whole, not only as a track, is not well understood by most individuals. No, so I would like to begin my talk with defining what biotechnology is. And according to the National Academy of Science and Technology, biotechnology is any biology-based technology which uses organisms or their parts to make new products or modify existing products, improve plant varieties, animal breeds, and microorganisms. So, simply, biotechnology is at the forefront of biology. So it could be a biotech can be a combination of biotech plus a semiconductor product, biology plus advances in chemistry, biology plus advances in artificial intelligence. So as long as your final product involves uh, a part organism or whole organisms, then it is within the realm of biotechnology. Now, biotechnology has two phases. The first one is the traditional biotechnology, which we are commonly familiar with based on its products. So it's definitely a product of fermentation. We have been using this for several years in our household. And then the second phase is the modern biotech. So the best two examples that I can give you with the current pandemic is the RT-PCR COVID-19 test, and then vaccine production, COVID-19 vaccines, which uh, can be produced in several ways. So, so what is the contribution of biotech in our university? So the first one is the creation of a biotech track. So as shown here in the slide are the major courses unique to the students who are taking the biotech track. Subjects such as medicinal plants and products, tissue culture, bioresource management and conservation, genetic engineering, computational biology, and bioinformatics. So these are unique only to those who would like to take biotech track. How about for the research? So for the last the latest batch that graduated 2019, I would just like to highlight the works of uh, two of my thesis advisee, Mr. Gilbert Sobrimisana and Myla Angela Soriaso. They did a DNA barcoding of rainbow sardines from selected marine habitats along Iloilo Strait. And this is the co-advisor is Dr. Pastor Jones Dinusta. So essentially what they did is they would like to figure out if the rainbow sardine here in Iloilo is similar to the one in the public database sequence. So in order to verify that, They did sample collection, morphological assessment, DNA extraction, PCR, sequencing, and bioinformatic analysis until they ended up with a phylogenetic tree like the one that you can see. So that is just uh, one of the works of the biotech majors. And then another one, this is a pre-med major, but they use biotech tools in order to perform their study. They isolated probiotics from tuba, and then they assessed whether any of these probiotics has an antibacterial property. So they did a minimum inhibitory concentration assay using rizazorin. They did that in a 96 well plate. And they also, once they were able to identify the active isolate, they identified that using 16S rDNA sequencing. So that's also involves the use of biotech tools. So what are the available resources that students can utilize to perform biotech research? So for many years, the Central Science Laboratory has been the main source, the main go-to resource of our students. Um, the staff here are very friendly and are able to accommodate our students. However, um, uh, because of the number of students and because of a specific need of the biotech, there's a need to perform the research using state-of-the-art equipment. So in 2018, I was able to secure a grant from CHED, and I put up the Biotechnology Research and Learning Center. So with this grant, I was able to purchase basic molecular biology equipment. We were able to do endpoint PCR and gel electrophoresis analysis of our isolated and PCR amplified products. And last year, or before the pandemic started, I was again able to secure another grant. And this grant was actually uh, allotted for the creation of a research lab for the College of Medicine. So in order to facilitate the administration and the operation of the biotech center and the proposed medical research lab, I I suggested to our university president to merge the two center, the biotech and the biomedical research proposed biomedical research center into one. And currently, we now have the Center for Biotechnology and Biomedical Research. So, a few weeks from now, the Biotech Research Center will be moving into its new location and merge at the College of Medicine. Okay. So, what is the current research activity in biotech? The modern biotech current research activity that is going on in the center. So at the moment, we are conducting genetic diversity of the rag native chicken using microsatellite markers. We are developing an automated workflow for COVID-19 RT-PCR test for high throughput testing. We have also done uh, previous studies, DNA barcoding of medicinal plants in collaboration with Dr. Gerard Penicilia, DNA barcoding of fish and shellfish conducted by undergraduate biotech majors. And the current capability that we now have by merging the two centers is that we are capable of doing sample preparation using bead beating. We can do low to medium throughput DNA, RNA extraction, either manual or automated extraction, DNA, RNA protein quantification, gel electrophoresis. We can do DNA barcoding of different organisms, gene detection using endpoint PCR, qPCR, RT-PCR, antibacterial, antioxidant, and DNA protection assay, plant extraction via rotavap and purification by HPLC, biochemical enzymatic cell-based assay using multi-mode microplate reader, and we do have provisions for bioinformatics software, commercial version, Genius SnapGene, and probably we're the only one in the university having an installation of math lab, but this one is being used by Dr. Alexander Balsumo. So the future prospect, what we would like to do in the next several months is to do gene cloning, perhaps DNA sequencing next year via Sanger sequencing or next generation sequencing, and quantification of genes by digital droplet PCR. Now, digital droplet PCR is is a quantitative way of measuring gene expression as opposed to qPCR, which is semi-quantitative. Now, what about our service to community? So, number one, thesis mentoring of biotech pre-med in micro students, undergraduate, master's level, and sometimes some PhD students. Consultancy for secondary, undergraduate, graduate, and faculty research. The lab has also provided a venue for conducting training in COVID-19 RT-PCR test last year. We also provide technical and resources support to WVSU Medical Center Molecular Lab. So currently, our PCR cabinet and some of our consumables were borrowed by the molecular lab. We have also provided cold chain storage for COVID-19 test kit and potentially COVID-19 vaccine. So I have received requests from the head pathologist of the molecular lab um, for a request to borrow the negative 80 biofreezer for storage of the Pfizer vaccine that will be donated by the Department of Health. So I guess that's all what I can say because I have a limited time. I hope I can uh, provide more enlightenment through your questions if there are any. Thank you and have a good day.